Very good evening, everyone. Please confirm if I'm audible and my screen is visible. Good evening, Sunshine. Good evening, Rahul. Am I audible? Can you all see the screen? Please confirm. In the chat box, please confirm. Okay, thank you for confirming, Rahul, Suresh. So I hope you all are doing great, doing good. So excited for today's session. Good evening, Ashu. Very good evening to those who are joining, those who have just joined right now. So I'm Dr. Tanushree and I welcome you all to another session of Brahma series. So far we have discussed the questions from Unit 8, the questions from the topics like probability, questions on Mendelian ratios, questions from subunit B of Unit 8, that is concept of gene, and extensions of Mendelian principles, that is subunit C of Unit 8. So we have discussed the questions based on topics like complementation tests, epistasis, and based on genomic imprinting and various topics which are given in the subunit B and C of Unit 8. Now today we will be discussing questions from Unit 8, and I'm sure you all know that Unit 8, it is about the gene mapping methods. So those who have missed the previous sessions, you can go through the go through the uh, our YouTube channel. So there the recordings are there. You can go through the previous sessions that I have taken of the topics from Unit 8. Now today I have taken Unit 8D, that is all about gene mapping methods, and gene mapping method subunit 8D is very important from exam point of view. Like from this particular subunit, you can expect the statement based questions. You can expect the numerical based questions. So usually the questions which are asked are based on the topics like the gene order. The questions are asked on linkage, linked, or how will you find out that it's a case of independent assortment, or it's a case of like, is it a case of independent assortment, or it's a case of linkage. Then from gene mapping, the questions can be asked on gene order, or it may be the question can be asked on distance between the genes, right? That is the distance genetic map or linkage map. And even from subunit 8D, the questions are sometimes asked on tetrad analysis, and also questions are asked on physical mapping, either based on deletion mapping method or somatic cell hybridization based method. So today particularly we will be discussing questions from these two topics from unit 8D, that is linkage and genetic mapping. So let's start with the questions. So I will be telling you what type of questions can be asked from these particular topics. What is the strategy of solving such questions? What is the trick you need to apply here? And even I will be telling you what all concepts you need to recall while solving a particular type of question. So here on your screen, you can see one type of question based on linkage map. So I want you all to go through it and try to solve it. Okay, and then I'll discuss the answer. So in a linkage map, the two genes A and B are 70 centimorgan apart. If individuals heterozygous for both the genes are test cross, the number of progeny with parental phenotypes, what will be? What will it be? Will it be equal to number of progeny with recombinant phenotype? Will it be more than the number of progeny with recombinant phenotype? Will it be less than the number of progeny with recombinant phenotype? Or it could be more or less than the number of progeny with recombinant phenotype, depending on whether the genes are linked in cis or trans respectively. So I want each one of you to try solving this question, try to recall what concept need to be applied here and how you're going to solve this question. So take your time. Okay, I can see some of you have already answered. So Achal, you are saying option 1, Ashu, you are saying option 2, Suresh, you are saying option 1. Let's see what others have to answer. Please try. Okay, it's very much important that you at least try, because these type of questions are usually asked in part B of the exam, and these questions are very direct. If you know which concept you have to recall here, you can easily answer such questions. So for this, you need to know what is linkage map, what is 70 centimorgan here represents, right? And what happens in case of linkage? How do you calculate recombination frequency? All that, all these points you have to recall here. Okay, Sunshine, you are saying option 4. So will it be equal? The parental phenotype will be equal to number of progeny with recombinant phenotype, or will it be more or less? Or it could be more or less both. Like, what do you think? It could be more or it can be less than the number of progeny. What do you think? Okay, Santi, option 1 you are saying, Sunshine, option 4. So basically, I would like to make you all recall about linkage. I have made a specifically this flowchart for you all for quick revision and for quick recall. So those who have attended my classes, I have discussed about these two topics in detail, like what is the difference between independent assortment and linkage. So I'm sure you all know about independent assortment. So Mendel, he performed experiments, and when he performed the dihybrid cross, he observed that the genes they sort independently, right? So I'm sure you all recall this particular experiment, breeding experiment, where Mendel crossed the round and yellow seeds with wrinkled and green seeds, right? So F1 progeny was all double heterozygous, right? And then on selfing of F1 generation, the Mendel's observed 9:3:3:1 phenotypic ratio in his dihybrid cross experiment. So in 9:3:3:1, the 9 resembles to one of the parent, that is, they were round and yellow, and 1 out of 60 resemble to another parent, which is wrinkled, and that is wrinkled and green in color. But other than these two, the new traits were observed, that is green and round, and also we observed yellow and wrinkled. So none of the parent was having these traits. So this indicates that recombination definitely would have occurred. So when we talk about the independent assortment, we say the genes assort independently, and we get new combinations in the progeny. And always remember, the basis of recombination is crossing over. So definitely crossing over has occurred, physical exchange of the chromosomes has occurred between the non-sister chromatids of homologous chromosomes, and that results into the formation, results into the recombination in the progeny. Now, when we are talking about linkage, it means that when the genes are located very closely on same chromosomes and they are always inherited together, then we say that genes are linked. That is, the chances of them to undergo crossing over is very less because they are very closely located on the chromosome. So it's become very difficult for any physical exchange to occur between these two genes, right? So they are always inherited together. So basically, when we talk about linkage, so linkage can be of two types, complete or incomplete. When we talk about complete linkage, it means that no crossing over will occur, right? That is, the genes, like for example, A and B located on the same chromosome. As you can see, the distance between them is very less, so they are always inherited together. So no crossing over, and in such case, when the gametes are formed, they will always be of parental type. However, in case of incomplete linkage, crossing over can occur, okay, because linkage is rarely complete. This has been observed through the experiments that we can have complete linkage. In complete linkage, no crossing over. But in case of incomplete linkage, the crossing over can occur. Now, when the crossing over occurs, then in that case, we get gametes. Some are parental type, some are recombinant type, just as that we get in case of independent assortment. So question arises how we can check for whether it's a case of incomplete linkage or independent assortment. So always remember, in case of independent assortment, the gametes formed are always in equal proportions. That is 50% parental type, 50% recombinant type. However, when we talk about the case of incomplete linkage, parental type will always be more than the recombinant. Okay, recombinant type will be less. So to test for linkage, we can perform test cross, the same that is mentioned in the question, right? In some of the questions based on linkage, it has been mentioned, or maybe in the exam, you can get a direct question that which test is usually being performed to test for linkage. So always remember it is the test cross. And what is test cross? Where the heterozygous is crossed with the homozygous recessive parent. So basically, linkage, we can say it's a deviation from expectations which were based on Mendel's principle of independent assortment. So this is the figure you can look here, that in case of no crossing over, like those who have not no idea about crossing over, I would like to show you here that how crossing over occurs. So just a quick revision about crossing over, that if like here you can see these are homologous chromosomes, and these are the sister chromatids, red ones, and these are the sister chromatids, blue one. But red and blue are non-sister chromatids. So you can see that when there is no crossing overs occur during the meiosis phase. Prophase of meiosis 1, it occurs. Okay, crossing over, it occurs in prophase of meiosis 1. And you can see that if no crossing over, then in that case, gametes form will always be non-recombinants. They will be parental type only. So here we have the parental combination A, B, and this in this form, right? Dominant alleles and recessive alleles together. However, if the crossing over occurs, like here you can see the crossing over occurs between this second and third strand, right? So you can see some part of the blue comes on red and some part of red comes over blue, right? So actually here, if I write down the combination, this one is exactly similar to parental type. This one is also exactly similar to parent type, because these two have not participated in the crossing over. But if you look at the combination of second and third strand, it will be, you can see dominant A but recessive B, and here recessive A, dominant B. So these are the recombinants. So this recombinants are produced definitely due to the crossing over. So as mentioned in the question, that we have to determine that if the genes are 70 centimorgan apart, what could be, like it is mentioned in the question, that we have to find out whether the parental genotype will be more than the recombinant, or will be less than, or will be equal, right? So here I would like to show you one more experiment, which was been a breeding experiment, which was being done. And here we are taking a comparative case, that is when the genes are unlinked, that is assorted independently, and when genes are linked, right? So here you can see that in case of independent assortment, as I told you, that we will get, depending upon which strands are participating in crossing over, we'll get two non-recombinant gametes and two recombinant. Like for example, here we are considering the case when of, like we are considering two traits. One is of the type of leaf and another is height of the stem. So the leaf can be normal or mottled. So mottled is a recessive trait, and height can be tall. Stem height can be tall, or it can be dwarf. So dwarf is a recessive trait. So here we are performing a test cross where a heterozygous is crossed with a homozygous recessive parent. And you can see in heterozygous condition, we get the dominant trait. So we are getting normal leaf and tall stem height. So during the gamete formation, like here, if we have the genes arranged right like this, so definitely, let's say if the crossing over is occur between the second and the third strand. So let me just draw the another homozygous chromosome. So if the crossing over is, let's say, occurring between these two, right? So talking about the gamete formation, this strand is not participating in gamete formation, so have the parental gamete only. Here, this one will also form the parental gamete only. But because as the second and third strands are participating in crossing over, this part of the chromosome comes over here and this parts move over here. So we'll get this type of new combinations, right? So these are the recombinant gametes, as you can see here. right? So I hope it is clear how the recombinant gametes are formed. So depending upon, now once the gametes are formed, recessive, homozygous recessive parent will only form the gametes with the recessive alleles. So basically, when depending upon which gamete fuses with the another gamete, so we'll get these four combinations. We'll get these four phenotypes in the progeny: normal leaves to all, motile leaves to off, normal leaves to off, and motile leaves to off. So here you can see that we are getting 50-50 per half ratio, right? That half are recombinant, half are non-recombinant. However, in case of incomplete linkage, what happens is if there is no crossing over, that is when there is a complete linkage, what will happen? In case of complete linkage, what will happen? We will get non-recombinant. 100% gametes form will be non-recombinant. But if, as I told you, linkage can be rare, so crossing over might occur, and in that case, similar as the gametes formed in case of independent assortment, same here the gametes formed will be half will be recombinant, half will be non-recombinant. But the catch here is that when the progeny is formed, the non-recombinants will be more in number than the recombinant. So here you can see 55, 53, these are the non-recombinant progeny. That is the ones which are having the parental genotype, and 8 and 7 are the recombinants. So you always have to remember that when the test cross is performed, the dihybrid test cross ratio is when the genes are assorting independently, we always get 1:1:1:1 ratio. That is all the four that we are getting here are in equal proportion, like 25% this, 25% this, 25% this, 25% this. So overall 50% non-recombinant, 50% recombinant. However, when the linked genes undergo crossing over, we get mostly the non-recombinant and fewer the recombinant progeny. So all these points are very much important for you to recall when you are solving such questions. So here, if you look at this question, the question is that in a linkage map, two genes A and B are 70 centimorgan apart. So first of all, they are not very closely located, right? Centimorgan, it is the distance which is measured between the genes. It is the distance between the genes is measured in terms of represented in terms of centimorgan in a genetic map. So if individual heterozygous for both genes are test cross, so which I showed you in the previous slide, right? A sort of test cross where we are crossing the heterozygous with homozygous recessive parent. So what would be the parental phenotype? So you have to find out whether it's a case of linkage or it's a case of independent assortment. So there, like always remember when we talk about this 70-70 Morgan, it is the recombinant frequency. The distance between the genes in genetic map is measured in terms of recombination frequency, and this measures the intensity of linkage. If the genes are very closely located, the recombination frequency will be zero, like there will be no recombination, no crossing over. And always remember in case of independent assortment also, the maximum value of frequency of recombination is 50%, because we know that when independent assortment is happening, 50% parental, 50% recombinant progeny we get. Now here it is mentioned that the genes are 70 centimorgan apart. So recombination frequency is more than 50%. So definitely it is not the case of linkage, right? And because it is not the case of linkage, so definitely we will not get the parental genotype more, right? So we can assume that it's a case of independent assortment where the answer should be option 1, where the equal parental phenotype equal to that of the recombinant phenotype we are going to get. So those who have answered option 2 and 4, Sunshine and Ashu, I hope it is clear why the answer here is option 1. Now moving to another question, the following figure represents a physical map and a genetic map for 5 different genes, A to E. Which one of the following statements based on the above is correct? So you have to study this particular figure given, and based on that you have to find out, by going through the statements, you have to find out which of the statement is correct. So here you can see that both the physical map and genetic map is given. So physical map where the distance between the genes is measured in terms of base pair, and genetic map where the distance is measured in terms of recombination, or it has been calculated on basis of recombination frequency, and it is represented in the measured in terms of centimorgan. So go through the statements and tell me what is the answer over here. Which of the following statement is correct? Is the region between B and C is more recombinogenic than other loci? Then second statement is in comparison to the region between A and B, the region between D and E is more recombinogenic. Then it is mentioned 1 centimorgan is equal to 1 kb. And fourth, it is if more markers were mapped between D and E, the genetic distance between D and E is likely to decrease. So what do you think? Which statement is correct here? Okay, so again, I'm getting mixed answers for this. Option 1, some are saying option 2. Okay, option 2 again. So I think there is a confusion between option 1 and 2. Yes, quickly answer the question. Okay, Madhumita, option 1. Rahul, you are saying option 2. Fine. Surendra, Sayanti, again, option 1. Surendra, you are saying, Sayanti, you are saying option 2. Fine. So see how you're going to answer this question. There is a figure already given to you. So you don't have to think out of the box, right? The strategy of solving such question is that always, if the figure is given to you, answer it accordingly. Answer the question accordingly. So as you can see here, we have the first statement is that region between B and C is more recombinogenic than other loci. So if you look at the physical map, the distance is 20 kb. But in the genetic map, it is only 2 centimorgan. 2 centimorgan means 2% recombination frequency, 2% recombination, right? So according to this, it's not correct. This statement cannot be correct. Then if you look at the second statement, it says that in comparison to the region between A and B, the region between D and E is more recombinogenic. Now again, if you look at the physical map, the distance between A and B is 10 kb. And again, the distance between D and E is 10 kb, right, according to the physical map. So based on physical map, we cannot answer this question. But if we look at the genetic map, then distance between A to B is 9 and distance between D to E is 20. So yes, in that case, we can definitely say that in comparison to region A and B, so A and B represents 9% recombination and D and E represents 20%, right? So yes, we can say D and E is more recombinogenic. So this could be the answer. Then option 3, it says 1 centimorgan is equal to 1 kb. This is incorrect because 1 centimorgan is usually equal to 1 million base pair. And then according to our statement 4, that if more markers were mapped between D and E, the genetic distance between D and E is likely to decrease. So no, usually when we talk about the genetic map, markers are being used in order to determine, like we use them to determine the location of the gene, but that is not the case that the distance, if the markers are mapped, then the distance between D and E will be decreasing. So therefore, statement 3 and 4 are also incorrect, and therefore answer is option 2 here. So those who have answered option 2, I hope it is clear that why it is option 2 here. So those who have answered option 1, I hope it is clear why answer is option 2 here. Yes? Are you able to follow, everybody? Please mention yes or no in the chat. Yes, are you all following? Okay, that's great, Surendra. What about others? So moving over to another question. Okay, thank you, Rahul, Sayanti, Suresh, for your response. Thank you, Sunshine. So moving over to another question now, that a test cross was made with phenotypically wild-type Drosophila flies having genes for sepia eyes and curled wings in heterozygous condition, and the following results were obtained. So again, a result is given to you, a table is given to you, and it is mentioned that how many are wild-type, how many are sepia-eyed, how many are curled winged, and how many are sepia-eyed curled winged. and the results indicate. So based on this particular table which is given to you, you have to based on this table you have to find out that this table indicates which result out of the four statements which are given. So first is that independent assortment as wild and double mutant types are almost equal proportion. Unequal segregation as it is showing departure from 1 is to 1 is to 1 is to 1 ratio. Complete linkage as the two genes are very closely placed. And fourth, linkage, the two genes are separated by 25 centimorgan distance. So what do you think is the correct answer here? These questions are very easy. Even this question was also asked in part B. So although while looking at the question, it might appear that it is very difficult, it could be complicated because a table is given to you, but it's not the case like this. Whatever concepts I have told you in the beginning, just apply it over here and you can easily answer such questions. Okay, Manisha, you are asking that anybody got 93% scholarship for Watson. So Manisha, I'm not the right person to tell you about it. You can ask it to CSTT. Okay, they know about the uh, like about the details of this scholarship. Okay. So yes, or even I will be giving you Manisha toll-free number. There you can call and you can ask about it. Okay, so I'll be giving you in some time the number, toll-free number. Fine. So yes, quickly answer this question, everybody. We need to discuss many questions. So faster you give me the answer, more we can solve the questions, right? So what do you think is the answer of this question? I always say try to omit options. Okay, four options are given to you. Try to omit options. Two options out of four. So independent assortment as wild and double mutants are almost in equal proportion. So first of all, you can see it is mentioned a test cross with was made with phenotypically wild type Drosophila flies having the genes in heterozygous condition. So you know that if the genes are in heterozygous condition, we get the phenotypic trait of the dominant alleles only, right? So here we are getting wild type. So this definitely indicates that sepia and curled wings are the recessive trait, right? So sepia and curled wings are recessive traits. So you have to consider alleles for it. And then you have to perform the test cross. So test cross means where you are crossing it with homozygous recessive parent. And these results were obtained. So if I'm considering this is for sepia eyes, then this one is for wild type eyes. Okay. And if this is I'm considering for curled wings, then wild type I'm writing it like this. So basically the cross has been performed where the heterozygous is crossed with the homozygous recessive parent, double homozygous recessive parent. So when such type of cross is performed, this is what we get the result. That 400 will be wild type, 150 will be sepia, 100 will be curled winged, and 350 will be sepia eyed curled winged. So parents, if you see wild type and the double mutant, so are you getting it in equal proportions? The thing is, are we getting it in the equal proportions? If it would have been in the equal proportion, then only the independent assortment it would have been. And one thing you all know that in case of independent assortment, it's a kind of a dihybrid test cross we are performing, right? So we should get 1 is to 1 is to 1 is to 1 ratio, but are we getting it 1 is to 1 is to 1 is to 1? No, we are not getting, right? So that means option 1 cannot be the answer. Then if you look at the third statement, complete linkage. If there would have been a case of complete linkage, we would have not got these combinations CPI and curled wings, right? Like if our parents are wild type and double homozygous recessive, that is CPI'd and curled wing. So how come we are getting CPI'd and curled wings separate individuals also? Right? So that means this is also not the case of complete linkage. Now you are left with second and fourth. Now definitely to test for this, like you have to calculate the distance. That means you have to calculate the recombination frequency. So how you are going to do it? So this is what I told you. So this kind of parent will form the gametes with the recessive allele only, right? And this type of parent can form four type of gametes, right? So we can get this combination, or we can get, so this is one combination, this is another combination we can get, or this is the combination we can get, or this is the combination. So different type of gametes will be formed, right? And depending upon when they fuse with the gamete of the another parent, what you will get, this is the result that you will get, right? So you will get wild type. This one, this genotype will give you wild type, which is already mentioned 400 individuals for it. Then curled wings. So here, although we will get for eyes wild type, but for wings will be curled, right? And here we, because the allele for the eyes are for coding for CPIs are in homozygous condition. So CPIs will get, and here we'll get CPIs and curled wings. So total progeny is 1000, right? And the formula for calculating the recombination frequency, please always remember, it is number of recombinant by total number of progeny into 100. So here our recombinance is 100 plus 150, right? Total number of progeny is 1000 into 100. And when you solve it, you will get 25 centimorgan. So this means that here, this confirms our answer, that answer is option 4 here. Is it clear to everyone? So out of 2 and 4, what is the answer? Option 4. So those who have answered 4, they are absolutely right. Moving over to another question. Double haploids are plants derived from single immature pollen and doubled artificially to form diploids. A double haploid population was created from F1 progeny derived from a cross between two parents, P1 and P2, one resistant and other sensitive to white rust. The parents F1 and double haploid population were profiled with a set of codominant markers, which is represented below. So codominant marker is a marker which is able to differentiate between homozygous and heterozygous genotype. Now, the following table summarizes the proposed percentage of 4 different types, 1 to 4 or double haploid progeny, assuming that DNA marker is unlinked to the gene governing the trait and linked at a distance of 10 centimorgan. Which one of the following options correctly represents expected ratio for unlinked and linked respectively? So here you have to find out whether A of unlinked is and second of linked is the answer, or A and 1 is the answer, or B and 1 is the answer, or A or 3 is the answer. So first of all, read the question carefully. I know this question appears to be little bit tricky, little bit difficult, but just recall what I told you, the concepts in the beginning, and you can easily answer this question. So let me know if you have any doubt. So what do you think is the answer of question here? So you have to analyze both. You have to analyze this gel profile, and also you have to see the table, and then you have to answer this question. Yes, what do you think is the correct answer over here? So here you can see this is parent 1, this is parent 2. One is resistant, another is sensitive to white rust. So F1 is the progeny, right? And then these are the double haploids, out of which you can see two are resistant and two are sensitive. So what do you think? So you have to consider, I'm giving you a hint, you have to consider two cases. One is for when the genes are unlinked and another when they are linked. So in that case, what is the answer? What do you think is the correct answer? So if you look at the unlinked, first if you look at the unlinked, you have to find out profile, like if you look at the profile A and B, you can see for 1, 2, 3 and 4, equal ratio is given, like equal numbers are there, right? 25, 25, 25, 25. And then for B, you can see 37.5 for 1 and 2, and for 3 and 4, 12.5, 12.5. So how you confirm whether it's a case of the genes when they are unlinked? Yes? If the DNA marker is unlinked to the gene, what ratio we should get in case of independent assortment? Unlinkage means we are talking about independent assortment, right? Okay, Ashu, you are saying option 3. Really appreciate you for trying. Sunshine, 50% recombinant. Okay. But here I'm talking about the ratio. So what would be the ratio? Recombinant and parental equal proportion, 1 is to 1 is to 1 is to 1, right? So first of all, you have to identify like these are 1, 2, 3, 4, which has been mentioned in the question, right? So if you look at the first one, it is resembling to the parent, this one, right? And if you look at the fourth one, this is resembling to the parent too, right? Now, if you look here, this is resistant, right? But resistant for resistant, we should get this kind of gel profile, right? The bands, we should get this type. But you can see we are not getting this type, right? So that means this one will be the recombinant, right? So this one is parental type, this one is showing parental type, but here this one will be recombinant. Then here, if you look at the third one, if you look, it is, you can see it is sensitive. So it should resemble to parent 2. But we are not getting the gel profile as given for parent 2, right? You can see this is what the bands we should expect. But this is what we are getting. So that means this one is also recombinant, right? And we know that if there is a case of non-linkage, or I should say when the genes are not linked or independent, they are independently assorting. So we should get one is equal ratio, right, for parental and for recombinant. So here out of A and B, A is correct. Okay. Now, when I say A is correct, so this is what you have to recall here, right? That in case of independent assortment, parental type and recombinant equal proportions. However, in case of linkage, if complete linkage, no crossing over, recombinants you get only in case of when incomplete linkage is there. And that two recombinants are very less than the parental type. Parental type will always be more and recombinant type will be always less. This is the thing that you all have to recall here. So when I say in case of unlinked, so when the gene and markers are not linked, the equal probability of producing all the four possible combination. So that means A is correct here, right? Equal proportion we are getting in A only. So that means answer cannot be option 3. So that means now answer is between option 1, 2 and 4. Then for linkage, I told you if you have to find out that which are parental, which are recombinant. So I already told you that these are 1, 2, 3 and 4, which I mentioned in the question. So this is resistant, this is resistant. So they should resemble to the P1. So here, first lane, the band's profile in this lane 1 is resembling to P1. But if you look at the second one, it is not resembling to P1. So that means this is having parental combination and this one is having recombinant. Right? So lane 1 is having parental combination, lane 2 is having recombinant. Similarly, if we look at 3 and 4, so the fourth one is resembling, it is sensitive. So it should resemble to parent 2. So it is resembling to parent 2. You can see the same gel profile, the band profile we are getting here, right? But for over here, if you look, we are getting, so this is recombinant, right? Because you can see it is not resembling exactly to the parent 2, right? This and this you can see. This is not the profile that we are getting for parent 2. Okay. So this is what we have to first identify. So here you can see lane 1, it is showing same band pattern as P1 and lane 4 same as P2. But lane 1 and 4, so that means lane 1 and 4 are parental and lane 2 and 3, these are the recombinants, right? And therefore, now it is mentioned in the question that the distance between the marker and the gene is 10 centimorgan, right? So if you look here, 10 centimorgan, so the proportion of recombinant will be what? Two recombinants are there. So 5 each, 5%, 5% each recombinant. And for if we have, let's say 100 parental and let's assume if we have to calculate the parental. So from the total, if you are removing the recombinant, you are only left with 90, right? And each parental will be 45%. So that is what is mentioned here. Like 1 and 4 should be parental. We have already mentioned here. So it means the value should be 45 for them. And for recombinant, it should be 5. Clear to everyone? So that means answer here is option 2. That is A and 1. Clear to everybody? Sunshine, I hope it is clear to you. If you follow the steps that I told you, first think for unlinked and then think for linked. Okay. And even when you are looking for linked, then you have to first read the band profile which is given, find out which are parental, which are recombinant, and then already it is mentioned in the question the distance between marker and the gene, that is 10 centimorgan. So proportion of each you can calculate. Okay, because this is the proportion which is given, right? Percent expected, percent of progeny with profile. Okay. So one of you has asked me about the Watson scholarship. So everybody, I would like to tell you that the deadline for applying for Watson Impact Scholarship is 31st July only. So you have very limited time left. So hurry up. Those who have not applied for it, please apply it, because you're getting a lot of benefits when you apply for this scholarship. Like you will get 1 year coaching for CSIR NET and GATE exam, and even for other competitive exams like QET, IIT JAM, GATB. Even you will be getting the access to the national workshops, global workshops which are conducted by Biotechnica. And even you will be learning about our programming internship with hands-on training and 4 certificate courses of your choice. You will get access to them also through this scholarship. And even you can always, like I know you have a lot of questions based on your career opportunities in a particular field and what to do after post graduation or PhD. So such type of questions you can ask to our experts. So through this scholarship, you will get the access to our experts on the WhatsApp. And even if you have any questions related to CSIR NET study or any the exams which I've told you, GATE exam or other competitive exams, any questions if you have regarding them, you can ask us. And then you will also get a pass to our CRISPR conference, which will be CRISPR conference that is named CRISPR Congress 2023. And also many of you, like we get a lot of queries, like many students, they request for the recommendation letter and industry references. So through this scholarship, you'll get access to that also, recommendation letter and 3 industry references. And even you will be getting the access to mentoring sessions and CV and resume designing and many more. So if you want any details about this scholarship, you can, like these are the toll-free number and where you can contact us. So now moving over to the another question. So the question is that plants with the genotype, so genotype of plant is given, were crossed and the double crossover progenies had genotype mentioned. So what is the order of the gene? So we have already discussed about the questions on linkage, and now we are moving over to the genetic mapping. So here the question is based on how to determine, like how will you determine the order of the genes. So parental genotype is given and double crossover is given and four options are given. So first of all, you need to understand that when we talk about genetic mapping is like when you talk about road map. So it's like where, or like Google Maps. So what does it tells you? It tells you about the location of a particular place, right? Similarly, when you talk about genetic map or linkage map, that tells you about the location of the arrangement of the genes and position of the genes on a chromosome. And you can the genetic maps are plotted by calculating the recombination frequencies, and the distance between the genes is measured in terms of centimorgan, or even in terms of a centimorgan, or even percent map unit mu also. And 1 centimorgan is equal to 1% recombination. So here you have to recall the strategy of how you can determine the order of the gene. So like you have three genes. So order of the gene, with this it means that whether the order of the gene in the on the chromosome is like MON, or is it MNO, like first M is located, then O and then N, or is it M and then near to M is N and then O is there. Okay, so like that you have to find out the order of the genes. So here, this is what I was telling you, that we can plot the map two ways, physical map and genetic map. So genetic map is where we are determining the arrangement of the genes and location of the genes on the chromosome, and it is plotted by uh, calculating the recombination frequencies. And as I told you, distances on genetic maps are measured in centimorgan or map unit, and where one map unit is equal to 1% recombination. So, like you have to find out, like here also some parental genotype and double crossover is given. So, question arises how you are going to find out the gene order. So, basically, parental combination is given and double crossover is this one given. So, tell me, what do you think is the correct order of the genes? Okay, Rahul, Achal, Surendra, Suresh, you are saying option 2. Okay, so I can see some of you have already answered this question. That's great. So what do you do to calculate to find out the gene order? So always compare one parental with any double crossover. Okay. So you have these two parental mentioned and these two double crossovers mentioned. So take any one parental and any one double crossover. So those who don't know how to calculate the order of the, how to find the order of the genes, always compare one parental with one double crossover. So here, let me take this parental and this double crossover. So this is already genotype is given, but that doesn't mean the order is this only. We have to find out the correct gene order. Okay. Now the trick here is always find the odd one out. Okay, like odd one out means, like for example, I give you these three figures. Okay, and I ask you which one is odd one out. So definitely you know that these two are triangle. This one is circle. So this is odd one out, right? Now here, how you will find out which one is odd one out? So I'm sure you know that the alleles which are represented with plus are wild type. We represent the wild type alleles with the alphabet and with the plus sign. And the one without the plus is the same alphabet, but without plus is the mutant allele, right? So if you look here, if you compare both the chromosome, you can see the M allele which is present over here, it is the wild type. But here, this is the mutant type which is present, right? Then if you look over here for N, this is the mutant type, right? But for N on another chromosome, it is the wild type, right? So here also this is wild and this is mutant. Now, if you look at the O, this is also wild type and this is also wild type. So if you look at the pattern is that on one chromosome wild type and on another chromosome, the same gene allele but mutant one is present. For N, mutant, another chromosome. On one chromosome, it's the mutant, another chromosome, it's the wild type. But for O allele, you can see in both the chromosome, it's wild type. So it's an odd one out, right? It is not following the pattern. So that means the correct order here is MON. So yes, Rahul, Achal, Surendra and Suresh, you all have answered the question correctly. Very good. So these are easy tricks, right? I'm sure these are very much helpful for you all, right? Just keep these points in mind when such type of questions are asked in the exam. Okay. Now moving over to another question, and here the question is about the distance between the genes. So here you can see it is mentioned that Escherichia coli was simultaneously infected with two strains of phage lambda. So phage lambda, it's a bacteriophage, bacteria eating virus. So one strain of lambda had a mutant host range, and it is temperature sensitive and known to produce clear plaques. And this is the genotype given. Another strain of lambda carrier, the wild type allele, so the genotype is mentioned for that also. The progeny phases were collected from the lice cells and were plated on bacteria. The following number of different progeny were obtained. So you can see some data table is given to you where some progeny phase genotype is mentioned and the number of plagues are mentioned. You have to find the order of the three genes and you have to also map the distance between them. And four options are given to you. So you can see here that is the order HCST or CHST or HSTC or HCST. And even the distance between is given, like whether the distance between H and C allele is 36 centimorgan or 15 as in first option, for second some other value, for third again you can see the values are same but arrangement of the alleles is different. It is HSTC, here it is CHST. So CNH 21, HNST 21 centimorgan, like that. And for fourth, it is H2C 36 and C2ST infinity centimorgan. So what do you think is the correct answer over here? Now again, to solve this question, there must be some concepts which need to be very, very much clear. Okay. So basically, if those who don't know, I would like to tell you about the 3-point cross. 3-point cross is where we consider the location of three genes on a chromosome and the cross between them. So we are considering the three genes here, the A, B, and C. So we have the dominant alleles represented with uppercase letter and the recessive allele represented in lowercase letter, right? So you always have to consider four cases. The first case will be when there is no crossing over. So when there is no crossing over between the alleles of the three genes, definitely we will get the parental gametes only. However, if the crossing over is, let's say, between A and B, okay, so we call it as single crossover one, right? So in 3-point cross, four possibilities are there. First, when there is no crossing over. Second, when there is a crossing over between A and B. Third, when there is a crossing over between B and C, okay, this is single crossover two. And the fourth one, where you have a double crossing over, okay, so this is A, this is B, and this is C. So that is what is mentioned over here. Diagram A, crossover between A and B, crossover between B and C in diagram B is shown, and diagram C showing double crossover, that is crossing over between A and B also and B and C also. Now you can see here that if the crossing over is between A and B, so obviously this is what the alleles, like physical exchange of the chromosomes will happen, and you can see this is what the gametes will get, right? So you can see the recessive allele A moves over here and dominant allele A moves over here. If crossing over is between B and C, so you can see the recessive allele C moves over here and dominant allele C moves over here. So this is what the combination we get. And if there is a case of double crossover, then you can see that these two are like, you can see that in this case. So double crossover means you have to consider both single crossover 1 and 2 both. Now to calculate the recombination distance between the genes, you always have to follow certain steps, and this is a flow chart I have made for you all, that you can always remember at the time of the exam. You can recall at the time of the exam that the strategy of solving such question is you always have to start with finding of parental and double crossovers, because I told you four possibilities are there, and if a data table is given to you, you always have to first find out which genotype is showing the parental and which is showing the double crossover. So always remember, parental ones are the most common and the double crossovers are the least common. This is the point that you all have to remember. So non-recombinant means parental. So always in the data table, whichever value, whichever number of the progeny is most common, that will be the parental, and whichever is showing the least number, that means they represents the double crossovers. Now, once you find out the double crossover and parental, then you have to find the order of the gene. Okay. Even if the order is given to you in the question, you have to find out whether it is correct or not. So order of the gene, like the question that we discussed in the previous slide. Okay. So like that, you have to find out the order of the gene. Once you find out the order of the gene, if the order given is incorrect, you have to rearrange the genotype. And then you have to find out the single crossover event one, calculate the recombination frequency for it. And the formula is that number of recombinant progeny by total number of progeny into 100. And then you will find the second single crossover, calculate the recombination frequency for it. And finally, you will plot the map with the frequencies. So those who have attended the regular classes, I have covered this particular topic in a lot of detail, right? Because this is right now we have a session of only 1 hour, so I can't go in that much detail. We are simply here understanding the types of questions asked from a particular topic, the strategy of solving, and any trick if it can be applied, right? So this is what is the flow chart that you need to recall while solving the questions. So tell me, what do you think is the correct answer over here? Okay, Rahul, Sunshine, Achal, Suresh, you are saying option 3. What about others? I can see some of you are not answering. I really appreciate if you please answer the question, because this is for your benefit only, right? At the end in the exam, you all only have to solve the questions, right? So are you liking the questions? Are you liking are you able to understand? Yes. Do you think, is it helpful? These type of questions and these type of discussion of the questions during these sessions of Brahma series. Yes. Are you enjoying the sessions? Yes or no? Okay. That's great, Rahul. Thank you. Okay. Thank you, Sunshine. Okay. Achal, Surendra, you are also saying yes. So I'm glad to see your responses. So, yes, what is the answer over here? Madhumita, okay, thank you. You are also enjoying the sessions. I hope it is helpful. That's great. Thank you, Suresh. Thank you, Ashu. So we have to follow the steps, right? So what are the steps? First is, what is the most common or most frequent genotype? So you can see 300 and 350, right? So these are most common. So that means these must be the parental, and least common will be the double crossover. So that means 4 and 6 will be the double crossovers, right? So once you identified parental, once you identified the double crossovers, the next thing is you have to compare, right? You have to compare. So I will take any one parental. So let's say I'll take this parental and I'll take this double crossover. Okay. So you can take any, okay. You'll get the same result. Even if I take this parental and this double crossover, I'll get the same result. Okay. Now, what do you have to look for? Odd one out. So you can see this one is mutant, this is wild type allele. This is the mutant allele of the gene on one chromosome, this is wild type. But here these two are mutant. So this is odd one out. So that means this must be in the middle. So gene order is HSTC and not HCST, right? So the next step is you have to rearrange, rewrite, okay? Otherwise you will make a blunder. You won't be able to find out the single crossover events, right? So according to the correct gene order, the single crossover event is the crossing over between H and ST, right? And the second crossover will be as the distance between ST and C, right? Now, what you have to do is, how will you find out which one is the single crossover event one? So for this, you have to write down both the parental. Okay, you have to write down both the parental. So here my parentals are H plus, HT plus, and C plus, right? And another is, so this is one parental, this is another. So this is H, ST, and C, right? So if the crossing over occurs between H and ST, what it will be? This one will come here and this one will come here. So what combination you will get here? H, St plus C plus, and here you will get H plus St and C. So where this combination is given. So you can see here that one is over here, H, St plus C plus, and another is H plus St C. So that means these are my single crossover event 1, and this rest other 2 will be single crossover event 2, right? 90 and we have 50, right? So how do you calculate recombination frequency? What is the formula? Number of recombinants by total progeny into 100. So that means for this you will write 86, 104, and even you will write double crossovers also, because always remember in double crossover, one of the crossover is single crossover, right? So divided by total progeny into 100, you will get 21 centimorgan. And similarly, when you calculate for the other one, you will get 15 centimorgan. So this indicates that answer here is option C. So I can see that this question, most of you have answered it correctly. So you can see the point is these questions are not very difficult and you can easily answer these questions if you know the technique of solving such questions, right? So I'm stopping here. I hope you find this session. This session was really helpful for you all. You have learned something new. And I hope like by solving the questions, you have gained the confidence, right? So just follow the tricks, just follow the strategies which I'm telling you, and definitely you will be able to solve all the Unit 8 questions based on these topics correctly in the exam. So this is all for now. Thank you everyone for attending the session. We'll see you in another session. Tomorrow, again, there will be a session at 5 o'clock. So please join. Thank you, everybody.